Okay, now it is time for meiosis 2. Yes, there is a second half. Yes, it repeats all over again with just a slightly different take. Okay, so at the end of telophase 1, we were, like before it splits, we had a diploid cell, right? We had 2 sets of chromosomes, like 2 sets of centromeres, right? We had twice as many chromosomes. And then once it splits, we now have haploid cells, okay? So we now have 2 haploid cells that we have to keep track of now, okay? Don't worry if they're not exactly circular, okay? So in prophase 2, we start the same thing all over again, PMAP, and now it's prophase 2. We have a new set of spindle fibers and centrioles appear, the nuclear membrane disappears, and the chromosomes remain double-stranded. Okay, so as opposed to in interphase where we duplicate before prophase, in prophase they just stay double-stranded. Okay, so I'm going to have my, we now have 2 sets of centrioles, which makes sense because we're going to split these cells in 2. Okay, and our nuclear membranes are disappearing. Okay, and inside, make sure I keep the same ones together. I have my yellow one on this side with a little bit of purple. And then I have my blue one with a little bit of pink. Okay, and over here I have my pink one with a little bit of blue, and my purple one with a little bit of yellow. Okay, and centromeres for everyone. Okay, so now we have our 2 haploid cells. Okay, haploid because we have split the 2 centromeres in half. Okay, and now we go from prophase to metaphase. Okay, so new spindle fibers, new centrioles, nothing else duplicates. Okay, metaphase, same thing as before. Middle, all the X-shaped chromosomes align. So let's say all the X-shaped chromosomes align in the center of the cell. Okay, and then the spindle fibers attach to the chromosomes. So 1 cell, 2 cells, centrioles at each end. Okay, and now I gotta draw these in the middle. Okay, so my blue over here. With a little bit of pink. And then my pink. With a little bit of blue. And if you want to draw it the other way, so the centrioles are on this side and the homologous chromosomes are lined up here, that's totally fine. It means the exact same thing. Okay, and my yellow one with a little bit of purple, and my purple one with a little bit of yellow, and then I always end with my centromere. Okay, and now my spindle fibers, because they have to attach to something, they actually are attached to the centromeres. Okay. Okay, so lined up in the middle, homologous chromosomes lined up in the middle. Okay, X-shaped chromosomes lined up in the middle. Centrioles on either side, spindle fibers attached to the centromeres. Okay, now anaphase 2. The spindle fibers contract, okay, the centromeres split, okay, and the X-shaped chromosomes get pulled apart. Okay, so now we have single-stranded chromatids are pulled to the opposite ends of the cell. Okay, here's where things get interesting. They already were interesting, but now they're more interesting. Okay, so cell number 1, cell number 2, okay, and I have my centrioles. Okay, and now I'm going to have my 1/2 of the X-shaped chromosome, so 1 chromatid going to this side, and then 1 chromatid going to this side. And make sure that when you split it, you follow what you have here, okay? So when I draw this, the yellow with a bit of purple and the blue with a bit of pink are coming here, and the blue and yellow are going here. Pink and purple going to this side, pink with blue, purple with yellow are coming to this side, okay? That's super important when it comes to this. Okay, so I drew my yellow ones. Now I'm going to draw my purple ones. Okay, and my pink ones. And my pink. With a little bit of blue. And my blue. With a little bit of pink. Okay, if you're like, hey, Miss Labiuk, how come over here this chromatid is pointing, I'd say you guess, to the middle, and now it's pointing away? And I just say that it's like the chromatids are being pulled to the opposite ends of the cell because they're not solid structures, okay? They're being pulled by their centromere. So that's why I just kind of make it look like it's being pulled by their centromere. Okay, however you make it look, works the same. Okay, and we obviously would have more. It wouldn't be just those 2, but for drawing purposes and learning, this is a good amount. Okay, so if I come up here back to metaphase, how many centromeres do I have here? I have 2. How many do I have here? 2. Okay, if I look here in anaphase, I now have 4. 1, 2, 3, 4. 1, 2, 3, 4. Okay, watch out. That's really important.